This is our second part in the lecture on DNA, the genetic material. So if you remember, in the first part we talked a little bit about the history of DNA, some of the research that helped discover what it is, the science behind actually starting to kind of crack the code of DNA, some of those players that were involved in what is DNA and how does it get transferred and moved and from one organism to another. And the idea of transformation, where the virulence is passed from one cell to the next. So what we want to start looking at now is, the DNA structure. Let's tackle this a little bit more, do a little bit more in depth on this, and look at how this strand is actually put together. So last time we talked a little bit about the basic structure. You got your little sugar house or sugar. You have your phosphate group, and then you have nitrogenous bases. And keep in mind, in DNA, we use adenine and thymine, cytosine and guanine. Those are the four associated with DNA. When we get into RNA, we're going to kick out thymine and put in uracil instead. So we'll talk about that as we get into the RNA information. But when we look at that DNA strand, the strand is considered or called a double helix. And we will have millions and millions of these nucleotides attached to each other with this thing called the phosphodiester bond. So the phosphodiester bond here attaches nucleotides to each other. So we've got one nucleotide here, one nucleotide here. And it's always the top part, or the phosphate group of a nucleotide, that will bond, this phosphodiester bond, will bond to the bottom of the carbohydrate, or the base of the house. So when we assign these little numbers, 5 prime, 3 prime, that is indicating where the phosphate is versus the base of the sugar. So the phosphate group is at the 5 prime end. The base of the sugar is the 3 prime end. So it's always stacked up where the 5 prime attaches to the 3 prime. And we have this orientation of 5 to 3 throughout the entire length of the DNA strand. So when we string these things all together, we get one side of the strand. We then take a string of nucleotides and put it on the other side of the strand. And the key here is this base will attach to the base that's on the other side of the strand. So an analogy to think about is an extension ladder. So you use a ladder to climb to the gutters, to clean gutters, or to change a light bulb, or whatever. The rung where you would put your foot is the connection from one base here to the base on the other side. The sides of the ladder where you would you put your hands on it and hold it as you climb, that is the sugar house and the phosphate chimney, this side of the DNA strand right here. So a very, very important piece of information was discovered back in the early 1940s, and this is called Chargaff's rules. So Erwin Chargaff was a Russian scientist in the 1940s that determined adenine bonds with thymine, cytosine and guanine, so ATCG. That is how the bases will pair on the DNA ladder. So if you have A on one side, there should be a complementary thymine on the other side. If there's a G on one side, there should be a C on the other side. So that was an incredibly important piece of information to discover to help scientists start to understand better the nature of the DNA structure. Now, Chargaff laid this foundation down in the early 1940s. About a decade later, in the early 1950s, a young woman named Rosalind Franklin was working on the DNA structure. Her claim to fame, what she should get credit for, is she took the first X-ray of DNA in the early 1950s. Now, in today's world, an X-ray is no big deal, but this is in the 1950s. The technology was nowhere close to what it is today. So she took this X-ray, identified the three-dimensional structure of DNA, and this is a copy or print of the actual X-ray, and this X shape indicates DNA is three-dimensional and it has a helical structure. So when we draw it out, the artistic rendition, it's got this kind of spiraling staircase appearance to it. That is the helical nature of the DNA strand. Take Chargaff's information and put that on top of that. If you have A on one side of the strand, it means you have a T on the other. If you have a C on one side, it means a G on the other. And this goes from the bottom of the strand all the way to the top. So what we're looking at here is basically a chromosome, piece of DNA. Now you can wind it all up into that tight little X, or you can unravel it into the chromatin, but that's a chunk of genetic information right there in front of us. So Franklin made an incredibly important contribution to the DNA structure with her X-ray. So it was 1950s. In the same lab, in the same facility, were two other scientists that were working on DNA. And these guys are named James Watson and Francis Crick. They cracked the code of DNA in 1953. So they get credit for the structure of DNA using the evidence from Chargaff, Franklin, and others. Now what's interesting about Watson and Crick is that they never performed a single experiment related to DNA. So they didn't do any research. They didn't conduct experiments. They didn't design this and X-ray it and work with it. But they took all this information that was out there and fit the puzzle together before anybody else could. So incredibly smart scientists, but just very interesting that they did not actually conduct an experiment to understand the structure of the DNA. So, but they are the ones who receive credit, fame, fortune, accolades, etc. about the DNA structure. So what they determined, DNA is a double helix. There are two strands. The two strands are polymers of nucleotides, basically long strings of nucleotides here. And it has the phosphodiester bond creating the backbone. So this phosphodiester backbone right here, 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 etc., etc., etc. The DNA strand will wrap around one axis, and then it is what's called anti-parallel. Now what this means is, let me put some arrows in here to hopefully make this a little clearer. Anti-parallel means one side's going to be going like this, the other side is going to be going in the opposite direction. So anti-parallel, let's rotate that, there you go. That is what's meant by anti-parallel. So while one side goes down, the other side's going up. So the orientation of the 5', 3' on this side is reversed on the other side. So kind of like teeth on a zipper. One set goes up, the other set goes down. So Watson and Crick were able to crack that part of the code and understand that information, which was incredibly important in giving us more knowledge about DNA. So here's a little bit better representation of what it looks like. We're not going to get into all the details about the master groove, the minor groove, that type of information in this course. Future courses in molecular biology and genetics, you will. But what we'll see is, here's the spiraling, or the helical nature. There's our ATCGs, the nucleotides that are base pairing, and the distances here are repeating. So this major groove distance is identical here, to the next set, to the next set, and so on and so forth. So there's a lot of physics behind how this structure is formed, and different bonds that hold the DNA strand in that shape. And if those bonds get broken, then we have problems with the DNA. So when we talked about genetics, we talked a little bit about Fragile X syndrome. Just briefly, it's a sex-linked trait. There's a problem with the distance in the major groove at the bottom of the X chromosome, and that leads to some big problems. Okay? So the double helix is a very, very amazing structure when we look at the shape of the DNA strand. Now, the replication process was a challenge for a lot of people. The Melson-Stahl experiment is what really cracked the code on this and helped figure out how it replicates. So one hypothesis was DNA replication is conservative. That's this diagram. You have your double helix, and then it just makes a copy of the double helix, and here's the original, here's a copy. That's known as conservative replication. The original always stays the original, here's the copy. Research showed that's not how it works. They had this dispersive hypothesis that here's a double helix, and what it did is it basically fragments, and new sections get added into the original pieces on both sides here. And you have, here's an original piece, a new piece, an original piece, a new piece. And it's basically all mixed up to create two new strands. Research showed that's not how it works. The Melson-Stahl experiment showed that DNA replication is what we call semi-conservative. This strand here will rip open. It will be broken apart and it will open up. When it opens up, one side acts as a template for this gold-colored information here. The other side acts as a template for the gold-colored information over here. That's how we get our two strands. It's a semi-conservative process. So each new strand is composed of half of the original strand, and then that continues again and again and again as it continues through the cell cycle. So that replication procedure is that semi-conservative replication. Okay, so what we're going to do is take a look at DNA replication. And actually, in the next lecture, I'm going to post a video over DNA replication. There's some really cool things out there on YouTube, some great animations that show DNA replication. Definitely make sure you're getting comfortable with what is required for replication, the pieces, the parts, the structures involved. So 3 things are required. Number 1, you have to have a template to copy. You have to have something to replicate. So that is the basic piece of DNA. Enzymes are absolutely essential to do this work. Enzymes are the workhorse of DNA replication. And remember back to food, and we talked about proteins and enzymes, and we talked about enzymes and pathways. Enzymes are essential here. They do all the heavy lifting of DNA replication. And then the third requirement are the basic building blocks, the nucleotides. You have to have nucleotides. So when you're replicating and adding new nucleotides in, you got to have those ingredients there, the basic raw ingredients to add to the replicating strands to make more DNA. So the good thing is, every time we eat anything, we're consuming nucleotides. Everything we eat is made up of cells. Those cells have DNA inside them. So we're taking their DNA and literally recycling their nucleotides and using it to build our own DNA strand. So if you had chicken this morning, all the chicken nucleotides, they're using adenine, thymine, cytosine, guanine as in nitrogenous bases. We're literally taking those and just plugging those into our DNA strand. So it's pretty cool. DNA is universal across all life, and it allows us to replicate it. Now, with replication, there are 3 general steps to replication. Step 1 is what we call initiation. Get it started. Trigger that process. Step 2 is the elongation of the replicating strand. So as you're copying it, you're copying more and more and more and more and more and elongating it. And then step 3 is what we call termination, the end of the replication. Done. Okay, so what we'll see is replication happens at lots of different locations on the chromosomes when we talk about eukaryotes. It's happening simultaneously because of the massive volume of DNA that we possess. In prokaryotes, it tends to start at one point, a single origin, and literally just goes all the way around the loop of chromosome until it gets back to the starting point, and then it's done. So we'll start looking at that in our next lecture. But what we'll definitely do as we go into the next lecture is I want you guys to check out the little video I'm going to post. It's going to take a couple minutes for the video, but watch it. It'll talk about the process, the procedure, and then we'll revisit that with the PowerPoint slides here. Okay, so this is what we're looking at, the basics. You rip apart your DNA strand, and you have the original template, and then here's a new strand on this side, the template here, and a new strand on that side. Okay, so let's check it out In the next lecture, let's take a look at how this actually works.